OK, so I'm going to talk about modeling network dynamics in hyenas using SIENA. But before we get to the hyenas, in my PhD, I studied the rock Hyrax. And we studied different questions, such as structural balance, the effect of the network on longevity. And recently, we found some interesting patterns about their mating networks. And that's one of the first networks I created for my Hyrax friend. And one of the first things I noticed is that it's pretty similar to the network of my Facebook friends. So here's the ultimate reason to study animal social networks. But in the tradition of this workshop, I'm actually lying to you, because my Facebook friends network is an ego network. So you don't see actually the whole connections of my friends except for between themselves. So to keep up with Dave's tradition. And then it was time to move on, and I wanted to work on network dynamics. So basically, moving from static to dynamic, what are the limitations of static networks? So one of them is that sometimes if we look at the static network, it's kind of only one sample of the social structure, and it may not be representative of the full range of social structures that are possible. And the second limitation is that it's a bit like looking at a picture and trying to understand how the painter painted this picture. If you look at one picture, you don't really see the process. You don't really understand what were the design principles. So moving to dynamic networks, you can follow how the network changes in time, and you can understand more about the leading processes in action. So I was looking for a system to study network dynamics. And I'm glad I can work with Kay Holcomb, who's been studying spotted hyenas in Kenya in the Masai Mara in the last 25 years. And Jen Smith, who's here, was part of this project as a PhD student. And so she's got a huge data set of like 1,500 individuals, about 100,000 observations of social interactions. And here you can see the effort invested in this project. This is Kay's effort, and this is my effort here. Yeah, but I did my part pretty fast. I don't know. It took a lot of time. Yeah. So the question I'll present here is what affects the network dynamics. So it's not about how the network affects its members. It's about the social structure itself. And I can classify, I guess, it to 4 categories. The first one is external factors, such as competition with other species, the abundance of prey in the case of the hyenas, and factors like rainfall and temperature. The second category includes structural parameters of the network, such as the tendency to close triads, the limited number of associations one can maintain, and so on. The third category includes individual preferences. So I can prefer to associate with individual of a specific sex, age group, or social rank. And the last category includes dyadic factors, such as relatedness and spatial factors. So I'll tell you a bit about what is already known about hyena society. So they live in groups which are called clans of up to 125 members. But these clans can be much smaller, and it mostly depends on the abundance of prey. The clans are composed of matrilines, so females stay in the clan, and they live up to 18 years. And so you can find in the clan up to 5 generations of females, their daughters and granddaughters, and so on. And males, when they reach adulthood, they will leave the clan at some point, and they will become solitary and will try to join a new clan and become immigrant males. And it's important to note that they can recognize their clan members using multiple modalities. So it's easy to, we can assume that they recognize their members. And it's, Darren talked about experimental manipulation, so it's an easy system to work with. If you want to manipulate it, you can get out of the car, and one of the hyenas will be happy to remove you from your social network. So the clan has a very rigid social rank hierarchy. So up here, there's an alpha female, and her matriline, so her daughters and granddaughters, are just below her. So when a new female, or actually when there's a new hyena that is born, he gets his rank just below his mother. And then you'll see the next matriline in rank and so on. And down below here, you'll see the immigrant males. So if you are a male and you join the clan, you start here. And you'll go up only when new males will join after you do. And you can go up only until here. You'll never go up in rank above any of the natal individuals. And that has important implications because when a hyena hunts alone, and that's actually done more often than not, so he'll get everything for himself. But if there's some company, and that happens a lot, they'll start conflicts, and the result of this conflict, the access to the prey, is dependent on the social rank. And it gets harder once there are more hyenas on the prey, and you can see that some of them may not be able to access the food. And that has huge implications on fitness. So here we can see the structure of one clan over 30 years. So in 1979, these are the females that were part of the clan, and this is the alpha female, and they are ranked here. And you can see that after 30 years, the proportion of the clan members that are descendants of this female is about more than 50%. The second female, her descendants comprise another like 25%, and the third female, and so on. Most of the females, actually, their matriline have gone extinct over time. But you can also see that this female here, F40, still got some descendants so that form part of the clan today or recently. So it seems like social rank, you can somehow compensate for your social rank. And we have some preliminary results that show that your social network can compensate for your social rank in a way. So social rank is very important, but perhaps it's not the only factor. And what we already know about network dynamics was done by Jen Smith. You can see that in her poster downstairs. So we can see that prey affects the network density. And in a period with low prey, the network is less dense. When there's more prey, you'll see here a lot more connections. And again, with low prey, the network loses connections. And the reason is that when there's less prey, the weakly ranked individual, the ones lower on the hierarchy, they have to go farther away to find food because they don't have access to what is found. So you will actually see less connections in the network. OK, so the first thing I did with the data is create this animation just to get an impression of the dynamics. So each kind of slice here shows 1 month of interactions. And you can see that the network is pretty dynamic, but you can't really understand what happens here and why it happens. So I'm using the SIENA method, which was developed by Tom Snyders in Oxford. And I'll explain a bit about it. I'm really glad to talk after Andrew, as he's given a very good introduction to ERGM. And basically, SIENA is very similar to ERGM with the addition of the temporal parameter. So it's an agent-based model, and we treat the networks as dependent variables given the first network we observe. It's a Markov process, so we assume that in each time step, individuals make their social decisions dependent on the current state, and they don't care about what happened in the past. I know that we all know that hyenas and other animals may have good memory, but I still think that most animals are kind of practical, and they care less. They care about what can make their life better now and not what happened in the past. But it may be like not so good at this Markov process for very short time steps. So in my case, I'm currently studying year-to-year changes. I believe we can live with this assumption. And the idea is that microsteps generate the network dynamics. So when we observe the network at year 1 and then year 2, we don't assume that the network jumped from one state to the other. We assume that many small steps generated these changes that we observe, and we kind of know that that's true. So that's another assumption we can live with. So in each of these microsteps, the model chooses 2 actors randomly, and they get an opportunity to change their ties. They can delete a tie, they can create a new tie, or retain a tie or a no tie. And the direction of change is determined by an objective function. So it's, again, like a logistic regression. And what we ask is as the response variable is, will I be connected to Dave in the next time step? And the predictors are the parameters we enter into the model. And so it's very similar to what Andrew showed. And the probability of change depends on a comparison of the change against other possibilities. So just to illustrate such microsteps, let's say that these 2 individuals were chosen, and now this is the objective function. So S are the parameters, and beta are the coefficients. And basically, this individual and this one ask themselves, will the network will be more attractive in terms of the model for me if I form a tie or not? So for example, if there's a tendency to be an isolate in a network, it may not be attractive for this one to form a tie. And yes?
What's a tie in the network here?
In the hyenas?
Yeah.
A tie, I'll get to it later. It's based on a gambit of the group method, but I have to binarize it. So yeah, I'll explain it. As with the RGM, we test for the goodness of fit for the model. So after we estimate the model, we simulate networks and we compare them, the model networks to the real networks. And this network, for example, does not seem very similar to this one. So maybe the model that we have does not really explain the process that we observe. OK, so how did I construct the network from the data? One quite big limitation of SIENA and of ERGM, I think, is that it's binary. So it requires binary networks. So I have to I had the association indices from the observations, and then I chose some thresholds in order to form binary networks. So I tested 3 different thresholds, basically the strongest ties in the network, and like 50%, so it's like medium plus strong connections, and then like the 75% most strong connections, so it eliminates only the very weak ties. So first, this one is not working. You just cannot model anything. It never converges. And I guess that it tells us that this is not really informative. You get very dense networks, and it cannot tell you anything about the process as the relevant processes. This one works a bit better, but the 75%, so only the strongest ties, works better than the others. So for now, I'll present only these networks of strong connections. Our networks are undirected, so they're based on observations of like coordinated activity. But you can actually use SIENA. SIENA was actually developed for directed networks. So if you have like grooming networks and so on, you can use it. And I'll present results for 3 networks I constructed, one that includes all hyenas, one that includes only the females, and one that includes only the males, because females and males may have different factors affecting their social decisions. OK, the parameters I used in the model, I didn't keep it simple. So we've got ecological factors such as precipitation. It's all averaged over a year. Prey per area, per square kilometers. Structural parameters include the tendency to close triads, the tendency to form a distance to connection. So it means that I am connected to Nina, but she's connected to someone else that I'm not directly connected to. The degree of alter, so do I tend to connect to highly central individual? It's the network degree associativity, and the tendency to be an isolate. There are individual parameters that include sex, social rank, and dispersal status, and each one of them actually has two forms. First is sex itself. Do I prefer males or females? And second is sex similarity. Do I prefer to connect to individuals of my sex or the other? And that goes also for rank and dispersal status. I should note that dispersal status is relevant only for males because they may be natal or immigrant, and females are all natal. And as a dyadic parameter, I've got relatedness. So basically, the question is, what causes the network to change from over the years? And I'll present results for 10 years, which I kind of divided the data into these chunks in order to keep it computationally possible. And it's also important to note that you can have like 10 years together only if you assume that the social processes are similar during these years. You can test for that in SIENA, but if you think that something really important happened in the last 5 years, so maybe you should model it differently and find different parameters that affect the dynamics. Okay, let's see the results. So one thing that we see here is that isolate is so the results are by color for all females and males. One thing that we see here is that for the female and male networks, the tendency to be an isolate is very strong. So just to remind you, since this is a binary network formed using a threshold, it doesn't mean that they are not really connected to anyone. It means that they don't have strong connections to any other males or females. And since it kind of masks all the other parameters, I'm removing it from the figure so we can see better the other parameters. So let's go over it. Prey has a very small effect, significant but quite tiny positive effect. So if there's more prey, there is a more tendency to connect. Precipitation has a negative effect. So if there's more rain, there's less density. And that is in agreement of what we already know. Usually when there's more rain, the prey species, they are much less clustered, so the hyenas have to go farther away to find them. As for individual traits, rank similarity is positive only for females. If you don't see any parameter here for all and males, it means that in the best model I had, this parameter was not part of this model, so it means that the model did not converge if I use this parameter, or that the goodness of fit was poor. Rank is positive only in the all network and pretty small. Dispersal status similarity is positive, so immigrant males tend to connect to other immigrants and natal connect to other natals. And dispersal status is negative, which in my coding means that basically immigrant males tend to form more connections. Sex is also negative, which again by my coding means that females form more connections than males. Relatedness is positive for the all and for the males network. So individuals tend to connect to their kin. We don't see it in the female networks. Perhaps because it is already being accounted for by other factors. Degree associativity is very small but significantly negative. So it means that highly connected individuals tend to connect to weakly connected ones, kind of preferential attachment, if you like. Degree of alter is actually positive for the all network and negative for males. So males actually prefer to connect to non-central individuals, but we see the opposite effect in the full network. Distance 2 is always negative, and transitive triad is always positive. And this is, it doesn't seem very strong, but it is something that I find in all the models I tried. So we always see this tendency to close triads and to avoid the situation where I'm connected to someone and he's not connected, and he's connected to someone else that I'm not connected to. And density, as is usually found in human social networks, is negative, and that basically means that the default result, the default decision is not to connect to everybody else. Okay, so there's a lot of results here, and what we can learn from it, what's new here. So we already know from previous studies that ecological factors affect the social structure. We already know that hyenas tend to form ties with their kin. I think that apart from testing all these parameters together, the most important finding here is the structural tendencies, the tendency to close triads and to avoid these unbalanced triads. And that is something that we already found in the rock Hyrax. So we found that these triads, the closed triads, are much more common than you would expect by random, and these ones are much less common. And that's also in agreement with what Andrew presented for the mannequins. And with Arik Kirschenbaum, who just joined us today, we kind of ask, okay, but why do animals want to close triads? Why do they care about it? What's the evolutionary origin of that? And we constructed a pretty basic model that says that if we assume that there's some benefit from cooperating with a number of individuals, and this number can be seen here, so it may be any number you like. So if I'm a hyena and it's beneficial for me to go hunting with another 4 hyenas, so it's efficient only if we construct these clusters. Because if I'm writing a paper with Dave, but half of the time he's cooperating with someone else, I lose the benefit of cooperation half of the time. I don't want that to happen. That's why it's unbalanced. And so the options for me are either to connect to Dave's friend and write the paper together, or just separate from Dave and work with someone else or work alone. Because if I keep this type of cooperation, I lose all the time. So we think that kind of very simply explain why we see this tendency towards closing triads again and again. So the next step would be to actually test how the network affects its members. So in the hyenas, we can test for changes in social rank, although it is very rigid. It does change from time to time. And we can test for reproductive success. And in terms of the SIENA modeling, it means that in each of these micro steps, actors will get the opportunity to change either a tie or a trait value. So I can actually test how a trait value changes, and not only the network itself, and see how the network affects its members. I'd like to thank my collaborators. Some of them are here, Eli Geffen and Adi Barokas from Tel Aviv University, and especially Kay Holkamp and Arik. And thank you.